Hello, so this is the first lesson on genes and patterns of inheritance. In this video, we're going to be looking at a few key terms to start us off, and then we're going to be looking at different types of variation, namely continuous variation and discontinuous variation. So firstly, we're going to look at some key terms. A gene is a specific segment of DNA whose nucleotide sequence codes for a specific protein. A locus is the place on the chromosome where a gene is found. An allele is an alternative form of the same gene which occupy the same locus. Gregor Mendel referred to these as factors. A genotype is the constitution of an organism comprising all the genes possessed by an individual, and the phenotype is what is observable or measurable in terms of that trait. Feel free to pause the video at any stage to reread these key definitions, and there's going to be more on the next slide as well. Homologous chromosomes are pairs of chromosomes which are the same size and shape that carry genes for the same trait. One member of a pair is maternal and the other one will be paternal, so one from the mother and one from the father originally. Homozygous are individuals who carry two identical alleles of a gene, and they are homozygous or homozygotes. Heterozygous are those who carry different alleles of a specific gene on their homologous chromosomes. They are known as heterozygous. The dominant allele is typically when the phenotype is expressed over the other one. So one phenotype will be dominant. In an example with pea plants, which we'll be looking at, capital T was the gene for the tall version of the pea plant's height, and it was dominant to small t, which was the dwarf version. Recessive is the type which is only expressed whenever both versions are present. The nature of variation. So millions of different species inhabit the earth, and it's pretty easy to tell the difference between different types of living animals, plants. Differences within a species, however, are a lot more subtle, and these differences are known as variation. So as we've already said, there are two types of variation. Characteristics which are controlled by a small number of genes and whose phenotypes are sort of clear-cut are discontinuous variation. These characteristics are qualitative in nature, with the environment having a very small or negligible effect. On the other hand, factors which are controlled by many different genes whose phenotypes merge together to give a continuous range of values are known as continuous variation. These characteristics are quantitative, in other words, they can be measured, and the environment will have a much larger effect. We're now going to look at some examples of both types. So our first example is an example of discontinuous variation. So discontinuous variation example here is the ability to or to not roll your tongue. So this is controlled by one gene, and it results in being able to either roll your tongue or not roll your tongue. We have a ratio here of 3 to 1. It's 3 to 1. And there's only one gene controlling this. Pea plants, another example we have here is they can be either tall or dwarf. It is a qualitative variation. In other words, you can describe it. You don't measure it with a ruler or measure it with scales. You can either describe a pea plant as being tall or being dwarf. You describe a tongue roller as a tongue roller or a non-tongue roller. Minor variations in height between tall and short pea plants will be due to the environment, and they're only negligible, very, very small. Another example we have here is with 3 different possible outcomes. So for flowers, sometimes we can get 3 different outcomes here. So in this example, whenever a red and white snapdragon are crossed, we end up with 1 red, we have 2 pink, and we have 1 white, giving us a 1 to 2 to 1 generation ratio being observed. The second generation phenotypes fall into, again, distinct categories, either red, pink, or white. And finally, another example here is our blood types. We have 4 different blood types shown here: A, B, AB, and O. This is somewhat simplified because we're ignoring the effect of the rhesus factor, just to keep it a little bit more simple. So again, we have discrete phenotypes, 4 in this case, and this is another example of discontinuous variation. Continuous variation is shown in this graph. There's a range of different heights, and many heights are shown. There is a bell-shaped curve here, which we can see, highlighting all the different heights in the human population. Most humans are found in the center of this graph, or around the center of the graph. So human height is a characteristic where there's numerous forms and they do not fall into discrete classes like we've seen before with discontinuous variation. The forms all merge into one and result in continuous variation. So something like height is influenced by many different genes and is also influenced a lot by the environment. The environment includes perhaps what you eat and your lifestyle factors. If we go down the middle here, we have the mean height for all humans, and most humans are going to be found in the middle section. So if we take 2 standard deviations either side of the mean, that accounts for the vast majority, around 95% of the population. This characteristic is quantitative. That means it can be measured, revealing a range of values with the largest number of individuals grouped around the middle. So this curve is known as the normal distribution curve. And this frequency is known as normal distribution. Characteristics like this result in a bell-shaped curve with some of the following characteristics. So the graph will be symmetrical, and the mean, median, and mode will all be equal to one another. Relatively few values will fall into the extremely high or extremely low categories. 68% are within 1 standard deviation of the mean. 95% of values are found within 2 standard deviations of the mean. And what that means is, if we draw the graph as such, we can see that 95% of the population are found in this green area of the graph. Due to being symmetrical, only 2.5% of individuals are found at the extremely tall section and the extremely short section. So this is the result of something known as polygenetic inheritance. So whenever there's continuous variation, the phenotypes are quantitative, and unlike discontinuous varying characteristics, you will measure them rather than count them. So here we have 2 graphs which we've already seen. We have continuous variation for height on the right and discontinuous variation for flowers and snapdragons on the left. You would simply count the numbers in each generation of colours for flower colour, whereas you actually measure the different heights of humans and find different values as a result of that. Many different genes control quantitative characteristics such as height. Qualitative discontinuous characteristics are only going to be controlled by a small number of genes, likely 1 or 2. So polygenetic inheritance is used to describe characteristics whose expression is controlled by many different genes and their alleles, with each allele making a small but measurable contribution to that phenotype. So that is our first lesson on genes and patterns of inheritance. Hopefully after this video you're able to look at the differences between discontinuous and continuous variation, and also hopefully you've took some time to learn some of the key terms which are going to be popping up from here on in.